Good afternoon, everyone. I'm Margaret Allman-Farinelli, and I'm the chair today. And Hiba Jaber is going to be my co-chair, and she's going to look after the questions. So I have the honour of introducing you to Andrew Holmes. I will say in this introduction, there are some words my mother taught me no lady ever says. So do take those as being in inverted commas. Andy has general interests in microbial diversity, evolutionary origins, and ecological applications. He first attempted a career as an agricultural labourer in outback Queensland, but then took the soft option of PhD studies at UQ, pioneering the development of 16S rRNA-based tools for ecology and evolution of pretty coloured bacteria. Finding that the lack of hard physical labour agreed with him, he spent postdoc time at the University of Warwick, UK, and Macquarie University on various topics related to microbial ecology and filling in a few links in the global meat cycle. He arrived at the University of Sydney in 2002, and after having been assigned the Unikey code of a hole, realised the future lay in gut microbiology. Andrew's current research has a focus on understanding the mechanisms of host-microbe interaction in the gut and management of gut microbial ecosystem for health. He's had numerous editorial roles with leading microbiology journals over the years and is currently a fellow of PSANS. He is located in the School of Environment and Life Sciences and leads the microbiome project node in the Charles Perkins Centre, where his title as Professor is a source of ironical amusement to all his colleagues. So a warm welcome for Andy.
Thank you very much, Margaret. So you've got the title up there. And what I'm going to do is give you a very brief bit at the start of some of the current and more recent projects that I've been involved in through the microbiome node of the CPC in particular, but also I'll touch a little bit, or at least have in the back of my mind, the things that I've done with Food Standards Australia, New Zealand, and also with the Food and Agricultural Organisation. In essence, the questions that you can see there tied to the various funding bodies are ones that come up recurrently for me in how we're looking at the relationship between the microbiome, diet, and a whole range of different applications. The one that I want to sort of emphasise, though, is this bit down here at the bottom. At the end of the day, for nutrition-related chronic diseases, it's the change in the food environment that's really driven these sorts of epidemics. And the sorts of questions that come up for that sort of thing are, how can we use microbiome data in predicting interventions or making disease diagnoses or prognostics for disease? But also, and where my involvement with the FAO and PSANS has come about, to what extent should we say that the way in which our foods is impacting our microbiome is something we should be taking into consideration for regulation, determining if foods are safe, or if people are making new foods that have got health claims, how the hell do we validate that they actually work? So a lot of what we do is aimed at getting at those sorts of issues. My way of acknowledging the traditional owners is to tell a little bit of a story about where I came from. So I hail from central Queensland on Durrumbal country, and I live on Wongal, and we're standing here at the moment in Gadigal land. But the picture that you can see here is the one I want to talk about. At a Australian Academy of Science event that was in Brisbane last year, we had a wonderful lecture from an Aboriginal panel, actually, but there was one person in particular where her real thing she was interested in was connecting people back to country. And one of the things that really struck me from that was the extent to which Aboriginal lands and their connection to country were driven by the plant foods that grew there. So a lot of tribal boundaries were actually defined by what plants grew there that they were then specialised at harnessing and managing. And that is kind of the theme for what I'm going to talk about today, is that resource availability shapes biological systems, whether they're human communities or the way that we're organising our society now. And for those of you who hadn't quite worked it out yet, that map is drainage systems in Australia. Major drainage systems often correspond with different biogeographic regions, and they align quite tightly with different Aboriginal territories as well. And Bidjara country is where you've actually got a confluence between the three major drainage systems of the east coast there. So it was a major trading site and also a contact point for different groups. And this is a wonderful panel. It's actually at least the width of this room, the full thing there that you can see up there. And it's unusual for central Queensland for having, these are nets that were used for trapping either eels, fishing, or other types that were there for use for catching small wallabies that were herded out. You can also see, quite unusually, a range of different hand signals, including one that looks suspiciously like a hang 10 thing. But it's one of the few sites where there's records of sign language that was, or at least this is what it's thought to be, sign language that was used in hunting. So anyway, with that digression and background there, let's get on to the real topic of today. The reason why the gut microbiome is so interesting to anyone who's interested in animal biology is that everybody poos, but that there are big differences in the food that goes into animals and the journey that it takes through that animal system before it comes out as poo. Now, that little table you can see there, I've just highlighted a few of the different animal models that we've worked on in various parts, and I'll talk through a few examples with all of that. But what I want to highlight to you is that all of those ones that we've worked with are essentially omnivores. And that means that you've got a mix of animal-based foods, which as a rule are hard to catch but easy to digest. Plant concentrates, and that's fruits, nuts, tubers, etc., where you've got storage polymers in the plant, so they're more energy dense than other plant parts and typically more easy to digest, certainly easier to catch. And then you've got the plant fibre, the bulk of the mass in most plant tissues, that's both hard to digest but easier to access. And what I want to emphasise here is that it's hard to digest, so it's slower to progress through the gut, and also that it fundamentally needs microbes for animals to make use of it, because most of the enzymatic repertoire that you need to break down those plant polysaccharides is carbohydrate active enzymes that are simply not encoded in animals, so they have to come from microbes. So here's three quick ones out of that same review paper that I foolishly neglected to cite here for you. Chickens is a current project with Sonia Liu and ARC Linkage 1, where we're aiming to improve chicken nutrition. Rats or mice is a major experimental model that we've used here with multiple collaborators at the CPC. And what we're really interested in the long run is being able to improve human nutrition. The bits that I've highlighted in green there are the parts of those gut systems where you've got the highest microbial density and where that breakdown of the plant material happens. You can see here that it's on the through route in people, parked off in a lay-by on the side in rats, and you've got two blind turns in chooks. So there's different ways in which the plant material ends up in the sites where you've got the dominant microbial processing going on. And that means that the relationship between intake and when the digestion actually starts to happen is different. And that has some interesting consequences for the periodicity of food intake, and things like time-restricted feeding and intermittent fasting play on those. Just to emphasise that on the rats, I've highlighted there the difference between immediately after a meal, where the cecum is quite small, very little in it, but the stomach is quite big. It was about 4 hours later when you can see this massive expansion in the cecum. So that's just to emphasise that there really is a time delay that's different for the easily digestible materials and their absorption into the body and what you see for the microbe-influenced ones. Okay, so now I'm going to give you a series of little vignettes on stuff that we've done over the past, well, probably 15 years now. I hope it won't be too overwhelming with too much information, but I want to make a series of simple points as we go through them. So the first one is that if you're an omnivore like us, then there's many, many different ways that you can assemble your diet. You've got an extraordinary range of foods that you could take. And one of the things that people noticed at the start of the obesity epidemic and these nutrition-related diseases is that diet can be sufficient to induce or trigger these diseases. And here's one that we did back in 2012 in collaboration with Mansour, where you can see a control diet with a typically low percentage of fat versus one of these experimental model high-fat diets. And clearly you can see major differences in the physiological state there, one of which is this dramatic change in gut barrier function. So gut permeability is one of the, let's call it a comorbidity, that goes along together with the obesity and the metabolic disease. And I'll come back to that one a little bit more later. So for a while, we did have this idea that diet was what it was all about. But what I want to emphasize here is that these diet-induced outcomes is not the end of the story. It's not just about food, calories, or even macronutrient distribution. And where I first got really excited and involved in the gut microbiome area was a series of papers from Jeff Gordon's group. And that figure that you can see over there is one of their more famous ones, where what they showed was that that classic high-fat diet-induced model for metabolic disease basically doesn't work in germ-free animals. So microbes also matter to these outcomes of diet-induced diseases. And that was the dogma for quite a while, until a rather surprising study, which hasn't got anywhere near as much attention as it should have, from a German group, came out about 2010. And they did nothing more complicated than say, hey, let's try a different mouse strain. And in these C3H mice, their high-fat diet totally upended the dogma that microbes protect, or absence of microbes protects against obesity. They actually had a worse obesity in the germ-free animals. And on a low-fat diet, they didn't see too much happening there. So genotype obviously also mattered to how the microbes might be involved. But they had this conundrum about the lack of reproducibility on the high-fat one, and it was pointed out to them by reviewers that, hey, your high-fat diet was not the same as their high-fat diet. And so we can now also see that fat type also makes a huge difference. And using a different type of fat to make these diets, they actually recapitulated the same result that the Gordon group had had. But they did that in parallel with their own version of the high-fat diet. And this was where there was something really interesting. They actually didn't get the same result that they had the first time. And it still was clearly different to what was seen with the other one. But what this emphasised, or at least their explanation for it here, was that the developmental history, whether it be epigenetic traits within the animal or the assembly of the microbiome, also has an impact on what the outcomes might be. So that led to a big debate in the field about, well, all these things matter, but what's the relative importance of genes, diet, and the developmental history of the animal? And the two sort of dogmas that sort of emerged from that were competing with each other. The first was that diet explains most. And this was a study where they compared two really quite different diets, a low-fat plant polysaccharide-rich diet on the right and a high-fat, high-sugar on the left. And there were 5 different mouse strains tested. And that's a plot of the microbial communities across those. And you can see that the microbes correlated very, very strongly with the diet rather than with the different mouse strain. So it was suggesting that in terms of microbe outcomes, at least, diet was most important. But then another study that came out was done in humans, following them over the course of, can't remember exactly now, I think it was a month or two, on, again, 2 quite different diets, one of which was a high-fat diet and the other of which was a high-carbohydrate diet. Each of the 10 series of dots of the same colour here is the same individual sampled at 5 time points on each diet. And what is really quite startling there is that despite the fact that each individual had significant periods of time on each of the 2 diets, the microbiomes very much correlated with individuals rather than the diets. However, here we've got real-world people that probably didn't totally obey their diet guidelines. Certainly we're having a much more mixed diet than what the mice were on their homogeneous one. And well, we think that they weren't inbred, but they were in part of America where that's possible. So all of that, when you put it together, means that we have to think carefully about what are the different factors that might be impacting either microbiome outcomes or what we ultimately care about most in the long term, which is the human health outcomes or animal health outcomes in the model. So how do we control that, more importantly, to be able to identify what the important variables are? So this was where I first partnered with Steve Simpson. It was one of his first big studies that was applying this geometric framework to the mouse model. So we had one inbred mouse strain, one climate with an SPF animal house, and then most importantly, one resource pool. So there was a fixed set of food components that were used to make up these diets. So we no longer had as much variability there, but we did have variability that could be experimentally tested in this geometric framework. So each of these series of dots that you can see in this bubble diagram here represents a series of individual mice that were on one of 10 different macronutrient distributions, so 10 different ratios of protein, carbohydrate, and fat. And where the individual animals sit on that depends on what their total energy intake was on those diets. So there were 3 different caloric densities in the feeds that they were given. And what that means is that we end up with a comprehensive representation of all of the possible ways that you can take those nutrients in. And our question was, if you had an animal on here, or here, here, or here, can we actually predict what its outcomes in terms of either microbiome state, from my interest, or animal physiological state are? So applying Steve and David's geometric framework, which most of you here will be familiar with, you can plot various response surfaces to see how they relate to the differences in intake. There's 2 simple messages I want you to get out of this without going through it in too much detail. The first is that there's no consistent relationship between nutrient intake and outcome. Every different parameter that you measure may have a different response to the way that the diet intake varied. And so you can see that for immune profiles, so it's lymphocyte ratios, glycemic control, or body fat at the top. With the microbes, I've only shown Bacteroidetes abundance here. It's one of the major dominant groups in the microbiome. And what you can see here is that it's highest relative abundance down the bottom here for protein and carbohydrate intake, which is basically this red region that you can see over here. But what I want you to also note is that if you take a slice through this 3-dimensional space in a different direction, so if you're cutting across horizontally, such that it's fat intake that you're looking as you go up here, you'll see that these Bacteroidetes, in terms of their relative abundance, really don't care about fat. For any one particular thing as you go increasing fat intake, there's no real change in Bacteroidetes' relative abundance. So the main driver for this main group in the gut was protein plus carbohydrate intake. And the main role of fat that we could see here was that it displaced protein and carbohydrate. It was effectively acting as a diluent. So then we wanted to look a bit more about how that protein plus carbohydrate intake might be driving the system and how that all operated. And now I'm showing you 2 groups. So Bacteroidetes and Firmicutes are the 2 main groups. So in all of us here in this room, you can pretty much guarantee they account for 90% or more of your microbiome. And the Bacteroidetes were primarily low intake specialists that looked like this, and the Firmicutes high intake specialists that look like this. So why might you get these different responses? One of the things that we hypothesized was that microbes can't live on sugar alone. They also need a nitrogen source to be able to grow, and differences in how they were accessing the nitrogen to support their growth on the carbohydrates might be driving this flip-flop. So the wonderful little pictures that you can see over here are NanoSIMS images. So that's nanoscale secondary ion mass spectrometry looking at the fate of labelled nitrogen that was supplied as an injection into the blood of the mouse under 2 different diet regimes. And what you can see really startlingly is that on these low intake regimes over here, that the bacteria in the gut are really intensely labelled with that nitrogen tracer. Whereas on the high intake regimes, they're basically not using it at all. So this indicates a real flip-flop in terms of what matters to the microbes in terms of their success. I don't have time to go into it here, but if you look here, you can see a lot more skinny bugs than you can in the top one. So not only was what they were using as a community different, which bacteria were most successful in there was different. And so broadly, the model is a bit like this, that animals recycle nitrogenous waste in ways that effectively fertilise the gut environment as well. So if you are eating large amounts of protein as an excess to your requirements, a lot of that protein is effectively shared with the microbes because it will also flow through to where they are in the gut. But if you've got a meat requirement level of protein intake, then over time, the nitrogenous waste still has to be disposed of. And it's essentially that processing that can be excreted back into the gut. So support of microbial activity in the gut under this more, let's call it low intake state of total protein, is actually got host excretions coming back in to support the microbial activity. I don't have time to go into all of that in detail, but I'm just going to point out that protein and carbohydrate that comes in your mouth ultimately leads to sources of carbon and nitrogen for bacterial growth. But the relationship between how it gets to the bacteria has got 2 broadly different routes, one of which is much more dependent on your own physiology. So now I'm going to go on into some stuff that we've done in a bit more detail that looks at how those insights can be exploited to understand the dynamic response of the microbial community to diet and how that might impact health in various ways. So first of all, I want to re-emphasize this idea that the ratio of protein to carbohydrate can act as a kind of a switch for whether you've got the high intake or low intake forms of microbes responding. Carbohydrate digestibility also matters because the carbs that are contributing to microbe growth are primarily the ones that are digestion resistant. So if you've got a more complex carbohydrate profile in your diet, a high fibre diet, then you're favouring microbial activity more strongly. Fat, I mentioned earlier, acts more as a diluent in our initial experimental models. So 2 big questions that we've got here are, to what extent does the carbohydrate profile matter in driving microbial response? And do we actually care about fat types with respect to the microbes? So as a very, very quick segue, this is another study that was done together with Len Storling. Here, we've looked at 4 different diets. One was a control diet, but we looked at 3 different versions of a high-fat diet. So in each case, 60% of energy came from fat, but there were 3 different fats, saturated fat, and then 2 unsaturated ones, either omega-3s or omega-6s. The only thing I really want you to note here is that even though the dogma is that microbes in the gut don't use fat as any kind of a nutrient source because it just doesn't support anaerobic growth for them very effectively, it did have quite a big effect on the relative abundance of hydrogen sulfide producing bacteria. And hydrogen sulfide is cytotoxic, and there was a very strong correlation between the level of these bacteria and the decline in gut barrier function that I mentioned in that earlier slide. The mechanism here is thought to be that the physiological response to fat in the diet is to release more bile to emulsify and allow its absorption. But the type of bile that's preferentially released varies according to the saturation level of the fats. And taurocholate, which is a sulfated bile compound, is the one that is in higher levels on those high fat, saturated fat diets. And that sulfur is what drives the increase in these organisms and sets up a feedback loop that impaired gut barrier function and drove chronic intestinal inflammation. So that's an illustration where you can get surprising consequences from something like fat that involve physiological feedbacks with our system, and their consequences for the body are potentiated by what microbe activity results from that. In most cases, rather than trying to do harm like that, what we want to do is to do good. And that means thinking about types of carbohydrate that support beneficial microbial activity. And this is that prebiotic concept. Now, in some of the work that I've done with FSANS, where we've been looking at ways to validate health claims, etc., after much reading and eye crossing and punching of walls, I realised that the vast majority of work that's been done in that space is actually nonsense. It's really, really difficult to find a study looking at prebiotic effects where the prebiotic was the only thing that was changed. There's usually so many different factors that have been changed that it's hard to ascribe the effect to this specific supplement. So we've had a look at our own version of doing that, where in a sense, we took native starch, which is relatively digestible, to contrast it to resistant starch, which is much less digestible, serving as a prebiotic effectively, and everything else was kept the same. So chemically, these were basically identical diets, but biochemically, in terms of their accessibility to microbes, they differed. And what we found was that there was quite dramatic differences in both the microbiome, but also in quite a wide range of physiological outcomes. Now, you can read those there if you look at. All I really want you to note is the red and the green are different. So there was a range of systemic differences that were a consequence of that potentiation of microbial activity. At the top, you can see something that was a little bit of a surprise, and that is that it also impacted those bile compounds. One of the reasons for that is that the resorption of bile, which is physiologically important for us, is dependent on microbial activity in the small intestine that deconjugates the bile and enables resorption. If you change the microbes, you change which microbes have got the capacity to recycle that, and you have these flow-on consequences for how fat might be impacting the system. But the one that I want to get you to focus on is one that's related to tryptophan and various neurological circuitry and also immune processes that can go on in the gut. Don't need to worry about too much other than that IPA is a tryptophan metabolite. And although it's ultimately coming from dietary protein, it's something whose concentration was really markedly changed just by doing this resistant starch versus native starch. And it's a great interest in a range of different things. But if you want to then ask the simple question, how do we predict which microbes might be producing this, and how might we relate those microbe changes to physiological consequences of that elevation of this metabolite, we start to see a challenge. This is what I'll focus on understanding this for the last half of the talk. And that is that those top 5 rows that you can see there are 5 individual mice. Each column is a different bacterial taxon. And you can see that overall, the resistant starch diets very strongly drove an increase in this IPA, but the amount varied a little bit from individual to individual. But more importantly, what you can see is that the correlation between a specific microbial species and the level of the IPA was a little bit harder to predict in a sensible way. The thing I most want to note here is that Bifidobacterium, which is the taxon that had been most implicated in this, was actually quite low in terms of correlation with IPA. But the main reason it was so low is that there were 2 different strains of Bifidobacterium across this data set, and they weren't evenly distributed. So you only saw a strong correlation of Bifidobacterium with IPA if you aggregated them into a higher taxon. Now, again, I don't have time to go through all the nuance here, but what I want to point out is that how you classify makes a difference to what type of correlations you're going to be able to see. And that comes into the way in which we apply these fancy machine learning algorithms to try and predict diet intervention outcomes in people. You feeling brains full yet? Okay, so I'm going all right. So a very quick summary. So I gave you a bit of detail there, but the simple message is that people differ in their physiological state and their lifestyles, including their levels of exercise and their diet. They also differ in what microbes have occupied their gut. The combination of what you ate, which is also feeding what microbes you have, means that those different microbes have different types of outputs in terms of small molecules. So this will include both the things that we think of as MAMPs, those microbe-associated molecular patterns that are the targets for the pattern recognition receptors of the innate immune system, but also a whole range of metabolites that are the target of GPCR receptors that inform endocrine responses, developmental responses, or they're just taken up as a nutrient resource, etc. So those different gut outputs give rise to different physiological outcomes. And they can include a whole range of things that more or less covers the range of things that are associated with our nutrition-related chronic diseases. What we really want to do is to be able to predict for a person who's presenting with one of these diseases what's their prognosis, and how might we prescribe a diet intervention or other treatment to reverse that. So this is an example from a relatively recent paper that gives you an idea of the broad strategy that's being used there. And basically, it starts by describing the system components. Stool is taken as a metagenomic analysis of the microbiome. There's a whole range of clinical data and patient data, including food frequency questionnaires and metabolomics. You look at what exposures they might have, environmental factors that might add on top of that. And then you do this multi-omics analysis to basically, in a sense, you're collecting everything and looking for correlations with outcomes. Now, what I want to quickly point out to you here is that if you look at this metagenomic analysis, there's two broad groups that people are characterising these as. One is taxonomic, meaning a list of what species are there, and the other is functional, which is a little bit of a shorthand way of saying genes and proteins and pathways. And there's been a lot of debate about what's the best of those, or most informative of those to use. I just want to emphasise that in the case of taxonomic data, across this data set here, they found 746 species across the full data set. But only 176 of those were really prevalent. If you said by prevalence, you said at least a fifth of people had them. If your idea of prevalence was more stringent and you said at least half of the people had to have them, it was only 95. And if your idea of prevalence was more than 90% of people had to have them, it was a very small number indeed. So the take-home message here is that you're going to have very sparse data. There will be a lot of zeros for microbial taxon relative abundance in any individual that you're trying to analyse by this. And as I emphasised in the previous slide, your answers to how many species there are will also depend on how you classify. So classification bias will also have an impact that exacerbates the complications there. So these big data sets are collected. The bit in the box there explain we conveniently ignore because it's too hard. They basically push them into one of these machine learning algorithms, and there are predictions that are based on the correlations that can be seen there to try and inform the response. Now, you might think, does that really work? And the answer to that is, it actually is small. I'll come back to that. It's on the later slide where I come back to that. The answer is it does work, but you really need to think about the interactions between all of these different aspects here. In the interest of time, I'm going to jump through Alison's bit here fairly quickly. But in essence, what this was doing was looking at how those carbohydrate profiles might interact with the protein to carbohydrate ratio and energy intake. So we had 40 different diets. Each segment of this wheel is one diet. And you can see straight away that we've got half of our diets have high protein relative to the other half. If we look at simple sugars, so it's basically sucrose in this study here, it also varies across those two domains. The resistant starch, or the more complex sources of polysaccharides, similarly varies across everything. And then we had a range of prebiotics, guar gum and inulin, that are not digestible by the animal system, but are accessible to the microbes that were on top of that. And we can map on top of those different diets various host outcomes. I want to quickly emphasize that if you look at something that's widely accepted as being a beneficial prebiotic to add to foods, resistant starch, and how it correlates with a fairly good metric of gut function, which is blood in the stool, you'll see that on these high protein diets, as you increase resistant starch, it makes things worse. On a high carbohydrate diet, as you increase resistant starch, it makes things better. So essentially, a prebiotic supplement is a challenge for the food industry because it's not necessarily going to be beneficial in all contexts. You need to think about the context, dietary context in which it's being given, because interactions can change the type of outcome that you get. Now, I'm not going to go through this because I don't have enough time, but I'll point out very quickly on this, these maps that you can see here are basically heat maps of 30 different mice that were given a subset of 12 of those 40 diets. And essentially, if you can see blue, it means we could not detect the microbe over 8 weeks all the way through. If you can see any of the warm colours, then it's increased in relative abundance on that treatment. And so essentially what you can see here is that for this organism, there were 3 cages of mice where it was just never detected, but in others it was. If you look at this microbe over here, you can see that it was actually very low levels, in fact, below detection threshold at the start in many of them. But as soon as they went to our experimental diets, it became detectable and it was strongly responsive to protein. So this illustrates 2 things. One is that zeros are tricky. A 0 at baseline isn't necessarily a real 0 because it might be capable of responding as soon as you add the diet component, or it might be like this, where it's a genuine 0 and it's just not there. Now that has big impacts for how we treat data in those predictive models, because you can't make the assumption of if a 0 means no response or conditional response without knowing that. So this is just to illustrate that if you include all individuals as a randomized clinical trial would, you actually get a much less predictive model and a wrong answer as to if you exclude those individuals that you had identified as being a genuine 0 and treat them differently. And on top of that, we've got other ones where they might be present at the start and they either disappear or stay, depending on who else is there with them. So our model that we came about with that is that individuality matters. So what you start with is important because each individual gut is an isolated island. What resources you've got available to the system matters, so you need to think about the diet context. But the community type or the interactions between the organisms also matters. So we need to think about what's the response unit in a number of different ways. The response unit could be a particular gene that gets switched on. It could be a particular species that's capable of replicating in response to a component, or it could be a network module of interacting species that collectively need to work together to do something. So to remind you, that was mice. There's also been some excellent studies that have shown that this is true of humans. This is one where these lower panels indicating variation in diet. These are people who ate nothing but those liquid foods as their diet for 2.5 weeks. So their diet was as homogeneous as one of those experimental mice, but their microbiomes still fluctuated a little bit from day to day. And more importantly, their microbiomes remained different from each other, despite the fact that their diets were identical. This is a typical person's diet pattern, and you can see that despite the weekend binge on whatever, the diet still remained reasonably constant, but also individually distinct. So response to diet is individual. And when we come back to this idea of these predictive models, we've got multiple things to think about. One is this, how do we deal with the zeros? But the other one is this network problem. Because we've got these individual microbiomes that are quite stable, we need to think about what are the response units going to be within that, and it won't always be species or genes. This is the one I thought I was going to flash up earlier. I don't want to talk about this too much, other than to say that this question of should we take the taxa or the functional approach is one that's been looked at. Here, the grey is data sets that are organised by taxon, and the blue is ones by these functional genes. It's pretty much unambiguous that taxon level information is more informative than just straight gene level information. Now, the corollary to that is that genome level. So if those genes were then assembled with a genome scale model, then that's operating a little bit more like a taxon one. But essentially, you do have to aggregate your information at the level of how bacteria actually grow as species populations. So gene is less useful than taxon or organism, but can we actually go that one step up and look at communities? So the two major genera or families within the Bacteroidetes are Bacteroides and Prevotella, and it's been repeatedly observed that they are actually bimodally distributed across human populations, and you get these distinct subtypes of communities. The third group is a number of different firmicutes. So we end up with these things that they refer to as enterotypes, dominated by Prevotella, Bacteroides, or firmicutes. And across a landscape of humans, our microbiomes would look something like this, where some of us would have a very much Bacteroides-dominated microbiome, some would have a very much firmicutes, and some very much Prevotella. But the challenge is that although this is now very widely accepted, it's hard to say exactly what the boundary is between them. Whether there's 2 or 3 enterotypes will depend on how far down the mountain you draw your separating boundaries. Now, why do they matter? Because those 3 different groups have very different strategies for how they access fibre, for how they form network collaborations with other microbes in the gut, and for what type of cell envelope they've got. So across those 3 keystone taxa, the type of signalling that you see with the immune system or with the neuroendocrine system will be quite different, and also the types of nutrients in your diet that might drive them will vary. Now, very quickly mention that we also see that these enterotypes differ quite dramatically across human demographic groups. And I just wanted to flash this one quickly because essentially you see Prevotella dominating in traditional societies that have still got a subsistence level nutrition, hunter-gatherers, communities, etc. In the modern industrial society like ourselves, the vast majority of us will have a Bacteroides-dominated enterotype. But you also see in less recent, so transitional societies, those that have not got a long history of an industrialised food supply chain, a different distribution of those two. Now, it's thought that this relates to the community assembly processes that start at birth. I really put this here just because I wanted to flag 2 recent PhD students that I've co-supervised together with Adrian Gordon, one of which was based in Indonesia, one of which here in Sydney on the Baby 1000 program. And essentially, these are still sort of ongoing to some extent, but we're looking at the relationship between maternal microbiome, the infant microbiome during the milk-fed period, and then what happens as you go on through the weaning period. And in essence, we have found across the 2 that the mothers have got this very, very different prevalence there. But interestingly, in both cases, they've now got, let's call it, access to an industrialised society's food for the infants. We're not seeing yet big differences in the infants, despite the mothers having that. I could stop here if you want me to, or very quickly talk about this one.
Wrap it up in the next couple of minutes then.
Okay. So what happens at the other end of life is also of interest. Many of you here will know about the Steve Simpson's Mega Mouse study, which followed the lifespan of mice in different diets. This is work that Zhen Bao did on that. Now, mice don't have the same enterotypes as humans, but we can still recognise two broad ways in which the communities have assembled when we look at the diet regimes that we've got. And here you can just think of them as orange and green. What I want you to note here is that the top row that you're looking at here is a high energy density diet. The bottom row is a lower energy density diet, and each column is either 6 months on the diet, 12 months, or 18. 18 is when you're starting to get towards old age for a mouse. And what you can see straight away looking at this is that the proportion of these orangey coloured ones is dramatically increasing as the animals get older. So this is indicating that with age, you are getting a change in the way that the microbial community is perceiving that environment, despite them having been on those same diets for all that time. And when you look at a distance-based redundancy analysis, the driver of this is really a change in how protein and fat are interacting with the carbohydrate intake. And I'll remind you here that one of those big switches that we identified earlier was the way that nitrogen is recycled within the gut to support the microbial community. So with age, does that change? We know that proteostasis is a feature of that declines in humans as they get older, so this is not an implausible proposition. Jen also looked at that in another cohort of mice where rather than a single time point, she followed them over time. We had a mountain of poo. And what you can see here is mice from this diet formulation 6 that mostly had the green type microbiome at 6 months, but were totally the other after 6 months. And you can see what that looks like in a transition over time in that animal there. There were some health associations, but I won't go into that here. Finally, for the benefit of those, I know Archie is one who loves Disney plots and things like that. We've now done a multi-omic analysis there, which lets you visualize what's happening in these aging mice in terms of the microbiomes here. In addition to the experimental diets, we also had a normal rodent brown chow. So the first thing I want you to note is that these are all the experimental diets down here, and this is the brown chow up here. So diet actually was driving the biggest differences that we saw between the microbiomes, but that shift between brown chow and those experimental diets was huge. One was a complex, unrefined, high fibre thing. The others were chemically defined constituents mixed together. But you can also see two clouds within these, so there are different outcomes being driven there. In the MOFA analysis, what you're looking at is subgroups defined by diet. So diet was having a strong individual effect on communities, but it was also individual mouse. So these are now clustered down below by what cage they were in. So you can see a hierarchy of different effects. But finally, when we look at the different informative factors, it's these ones that are coloured on the side here. There are a number of factors that we were able to identify that really were specifically associated with age. So the age of the animal was driving its microbiome response to diet. So age does matter. I can't yet really tell you exactly how it matters for health outcomes, but I can tell you that it potentiates the way in which diet and microbiome interact. So I'll leave that there with my favourite group photo, even though it's a bit out of date now, because that's the Bristol school chart and chocolate cake in the bottom.
All right. Do we have any questions?
I can just, okay. I think because of the Zoom recording.
Yeah.
Thanks, Andy. It was great, like always. I was just wondering, it's so interesting to see rapid and dynamic changes in the microbiome across time scale of an organism. So the microbiome is same, their division rate, their life cycle is same, but when we study it in mice, that has a different age span, and when we study it in human, how the dynamics would change on an organismal life scale and how you will compare it on an evolutionary
Yep.
scale?
Oh, I guess the simple answer is yes, they change. You've got, it's not just so the temporal dynamics are important. So ontogeny varies, which can have actually, I think I've got one down here. Yeah, so pigs is another one that we've looked at. So pigs is probably, particularly as the immunology thing. So there's a quite well understood ontogeny of immune development in pigs because they're precocious, and so it's really easy to get a germ-free piglet and then model most of that. And for pigs, the maternal immunoglobulins all come in basically a 3-day period straight after the piglets are delivered, and they're mostly via the sow milk, and they're absorbed. And then the piglet's immune system develops much faster than humans, and so you see maturation by roughly 1 month of age. And what you can see, and this is old school here now, but at 7 days of age, this doggy fingerprints over here, basically you can see every piglet had a different microbial community. But at 21 days, in this narrow little window, suddenly piglets that were co-housed in the same cage had more or less identical or highly similar fingerprints. As we went on a bit longer, out to 36 or more days, you started to see a little bit of divergence again. But that window correlates very closely. So where you see a similar thing here to the transition point between the loss of the maternal and the onset of the developing immune system. And we think it means that there's a window in there where defenses or innate processes are more actively filtering, so that the communities are quite closely shared. So I think that kind of answers your question in the sense that, yes, there are differences between all those different animal systems. But when you look at what the processes that are controlling the microbial response are, it is often in most cases, it is possible to identify ways in which you can draw those extrapolations or generalizations across them.
I'll just check if there are any questions from anyone online. Are there people online? No. Okay, that's fine.
Thanks, Eva. Thanks very much, Andy. But for those of us who don't work in this area, the complexity continues to be daunting. I was struck by however, by the simpler breakdown that you described for us between the Bacteroides and the Provotella and the third group of organisms. And I'm just curious to know whether where that analysis is done, whether it's predominantly environmental factors, such as whether you grew up in Malaysia or in the United Kingdom, or whether there actually are human post-genetic factors that are relevant here.
That's a great question. There's been a lot of debate about whether there are human genetic factors that really determine microbial colonisation. A lot of that's been done out of twin studies. I think it's probably fair to probably still be Christensenella would be the only one that there's any real half meaningful evidence for that. I think for the other several 100,000 microbe species that you find in people, we can say that it's more environmental acquisition. Now, that doesn't mean that there's not going to be species specific or family unit things, because your exposure is going to be dominated by who's immediately around you. So you do see maternal, in particular, associations. It should be better phrased as maternal or closest carer, depending on what they are. In terms of being able to draw generalizations, in microbiology, we distinguish strains from species, and then so the species complex in microbiology intrinsically says they're not all the same. They're just usefully similar for the purpose of lumping them together, and you will find extraordinary strain variation amongst everyone here. But at the species level, you can identify some generalizable features. So Bacteroides, for example, generally have large genomes and can be selfish. They're very, very good at utilising starch, and their biosynthetic capacity is such that they don't need to collaborate with anyone else. So that Bacteroides enterotype is really dominated by just that one species because they don't need anyone else. Prevotellers have small genomes, often deficient in biosynthetic pathways for amino acids and vitamins, and tend to form collaborative networks. So it's a different style.
I'll ask a second question if I can.
There's one question.
No, you take it. Thank you.
Hi, Andy. I can.
It needs a bit of too.
Hi, Andy. Just a question. I'm always intrigued how robust the human microbiome is to diet. Much more as you've shown than the mouse microbiome. I'm just wondering if the plasticity of the microbiome is much greater in humans in early ages or gestation, if people have shown this, if we should be targeting young kids with changes in diet, if we want to be are we too old now? Is our microbiome so established that we can't do much?
Yep. It's another great question. There's a number of issues around that. The first one, in that, let's say the very young versus the adult, is that we really should think of them as two completely different microbiomes. They're temporally related, but the diet and the consequences of postnatal development mean that the environment in an infant is very, very different to an adult. So the milk fed is one story, and then you've got the transitional period where you complete the postnatal development and complete weaning. And to the end of that, so by the time an infant might reach 3 years old, the microbiome is much more variable than the examples that we saw here. And that's largely a consequence of continual fluctuations in both the host side through development, and the diet tends to be a bit more up and down as you go through there. The idea that it's actually, you'll often see the microbiome pitched as being a good intervention target in health because it's more manipulable. As you just kind of raised, it's not as manipulable as we might think, but it's also not necessarily as stable as we might think. When you do those contrasts between the mice experiments, they're actually really pushing the system. They range from 0 fibre to high fibre. If I put you on a 0 fibre diet, it would do strange things to your gut in a relatively short amount of time, and we would see a dramatic shift in the microbiome. So when we put people on diets, they're still maintaining a reasonable amount of fibre, that's when the microbiome is quite resilient to disturbance. So it depends how hard you push it. Yes.
All right, we'll take one more question and then it will be time for lunch.
Thank you. And just actually a follow-up comment to that. So if you do some of those diet experiments in mice that have more like wild microbiomes, so more diverse microbiomes, they don't shift as much as the inbred like SBF microbiomes do as well. So when they're closer to humans, the microbiome behaves a little bit more similarly. But my question was actually, so you talked about how looking at the microbial taxa was more predictive than looking at the functions that microbes can perform. But I guess that's being measured at the level of what they can potentially, what potential functions they have based on their genes. So do you think if you measured what they're actually doing, so for example, how much of given metabolites they're producing, that would be more predictive of some of these health outcomes?
Yeah, the way that I think about that is what's the response unit? So a gene or a piece of DNA doesn't actually have any response. It encodes a protein, but the protein also won't necessarily produce something unless it's in a cell that survives, that provides the cofactors and the reactants, etc., to do that. So a metabolic pathway is, again, one higher level of organisation. That's where you really are at the level where there'll be a correlation between a metabolic pathway and a metabolite that might be outside the cell. So that's moving from the original functional, which is just a gene list, to a pathway list. But in terms of diet response, it's not just expression levels of pathways that dictates activity, it's also the numbers of the cells. So the cell is the actual response unit and they replicate and grow. So it's the change in the population size of those in conjunction with what pathways are there. And so that's where you move to lumping things together as a species or a set of cells that are the same is a tighter correlation to the activities. I hope that's answering your question, that essentially what you're doing is you're aggregating the information at levels that more closely relate to how the total output will change in response to diet.
All right, we might just leave it there. Thank you, Andrew, for a fascinating talk. We do have a light lunch just outside, and I'm sure Andrew will be around to answer any more questions because I know there are a few we didn't get to. If you're new to this seminar series and would like to join the mailing list, we will have a piece of paper around. You can just write down your email address and we'll add you to that list. Is there anything else we need to remind people of? No, that's all. All right, thank you.
Thank you very much.